Good morning, everyone. Welcome to the Structure Biology Breakfast Workshop today. We appreciate your effort in getting up early this morning. My name is Wei Jing. I'm in the Omics Vertical Marketing Structure Biology group. I'm personally doing the native MS charge detection mass spec. I also have two other group members, Yu Qixi and Yi He, hiding somewhere in the audience. They are in charge of the HDX mass spec and cross-linking mass spec. Today we will have three great talks aligned. And our first speaker is Malvina Papanastasiou. Malvina holds a group leader and research scientist position at the Broad Institute of MIT and Harvard, while she is leading structure proteomics in the CAR group. She is currently a vice chair of the International Society of HDX mass spec. She has more than 15 years experience in the field of HDX mass spec, advancing technologies to better understand protein structure and function relationships. Let's give the stage to Malvina.
Thank you, Wei Jing, for pronouncing my name correctly. And thank you all for showing up. I am Malvina, I'm at the Broad Institute, and I'm really happy to talk to you today about recent advances we have in our group at the Broad Institute in the HDX MS space. There has been a slight change of plans, so even if you're here for cross-linking, this is what you get for showing up. We have been... Okay, lovely. So I am in Steve Carr's group. We do much of proteomics, but we are very happy because we are getting inspired by advances in this field, and then we take them and we apply them to HDX-MS, to advanced technologies there. So we have been doing HDX-MS for many years now, and we have a big application space that I am showing here. There is no laser pointer, so I'm just going to have to explain. For those of you who don't know, HDX-MS is an excellent technique for probing protein dynamics, protein-ligand interactions, and protein-protein interactions. Some key advantages that you have is that your protein is in a native state, so you have it in your buffer of choice. The analysis is fast compared to other high-resolution structural approaches, and it's particularly attractive for very dynamic things. So think of IDRs, IDPs, dynamic proteins, and also membrane proteins that are difficult to get with high-resolution structural approaches like X-ray and cryo-EM. And in the past year, we have been working really hard to advance the technology, and we are super exciting because now we can apply it also to mega Dalton scale complexes. And please go and listen to Shonak, who is giving a talk tomorrow in the HDX sessions to show some beautiful data we've got from like really large complexes. So how is the workflow? How are we doing the experiments? I'm going to go a little bit into this. We have the protein in native buffer, that's the equilibration step, and then we do a fold dilution with deuterium, and the clock starts ticking. So typically in these experiments, we perform a time course. So we want to probe the protein at early seconds all the way to a few hours. We can select the temperature at which we make these experiments, depending on the protein we are investigating, etc. At the end of each time point, we quench the reaction. And what do we mean by this? We drop the pH at 2.5 to slow down the exchange process. And also, we drop the temperature at 0 degrees Celsius. So all of our analysis occurs at 0 degrees Celsius. We then inject the sample, and we do a very brief online digestion. We have immobilized proteases, and we do a 2-minute digestion at 200 microliters per minute. And then we analyze the data to keep it simple in MS1-based mode. At the very end, what we end up with is this D-uptake graphs. So on the x-axis is the time. On the y-axis is the de-incorporation. And what does the de-incorporation tells us? It's about the hydrogen bonding network of your protein. So what is solvent accessible? It doesn't participate in hydrogen bonding, will exchange fast. What is in the core of the protein, participating in secondary structure and interaction, it will exchange slow. So all these uptake graphs, then they are correlated with protein structure, interactions, and conformation of the protein. So this is a cartoon of the workflow we have at the Broad. We have a fully automated workflow, and we like to call it high throughput. I know for some proteomics people, this is not really high throughput, but given how the field was 10 years ago, we consider it to be high throughput. So you have your protein of interest, and assuming that you're doing a differential experiment, you want to find where your ligand binds. You have your protein in DMSO, your protein with a ligand, and you put your vials in the robot, and the robot will automatically label the samples, quench the samples, inject them, digest them, etc. Again, at 0 degrees Celsius. Then we measure the profiles by mass spectrometry, the deuterium uptake profiles, and then we analyze the data. So with the sensitivity that I'm going to show you that we have these days, we leverage very short gradients. This is something that I didn't mention in the previous slide, but in order to maintain the deuterium label, you have to do fast chromatography. So typically, you have a 30-minute window to finish your analysis of each sample. We do it in 6 minutes primarily, for proteins up to 300 kilodalton at least. And this allows us to do 72 injections per day. And we have a fast turnaround of results. So what we do typically for projects where we have multiple ligands, we operate what we call in a screening mode. So think about 72 injections in a day. And then that could be like single-shot injections, or it could be with triplicates, and to get some statistical analysis, etc. And then you ask the question only at this mode, does my drug bind? Yes, no. If yes, then what you want to do is select drugs and go to the conventional mode analysis and do a proper time course to better understand the effect of drug binding on your protein dynamics. So depending on the projects that we have, we use either mode or both. Okay, so those of you who do HDX, you know what I'm talking about here. Those of you who don't, please allow me a minute to introduce how we do these experiments. Most of the field utilizes microflow configurations. And why is this? Because the flow path that we have in the systems that we use, the void volume is very large. It's like 60 microliters, 65 microliters. So in order to allow for the fast elution of the peptides, you need to utilize high flow rates. So we are in a regime where we're talking about 20 to 80 microliters per minute. And if you do this, of course, you take a hit on your sensitivity. I show the TIC graph at the bottom there, if you can see. And then if you inject the QC standard in our lab, which is a jerked peptide digest, you see that for short minute gradients, you get around 2,000 peptides. But if you prolong the gradient, this does not boost sensitivity because you are already capped on that. So we have been thinking, how can we make the instrument more sensitive, because proteomics has done that for years, why not us? So Shonak here has been working really hard over the past year to optimize 2 different configurations that hopefully, depending on the system you have, you can employ them. One configuration, he was able to minimize the volume to 8 microliters per minute that we call the capillary mode. So you can here employ flow rates within the 3 to 5 microliter range. And then he kept on going, and we also made the nano version in which the void volume of the system is less than 2 microliters, so now we can apply really flows in the nano flow regime. And if you see the TICs at the bottom, I hope you can see it clearly, the gain in sensitivity we have. We are an order of magnitude higher, even in the capillary mode, and it looks only better in the nano mode. So how does this translate into jerkets? If you see now the graphs at the bottom in the capillary mode and on a 6-minute gradient, you keep getting a boost on sensitivity, and it's only becoming better at longer gradients. Now, if you have enough material, great, just go and inject the same amount, you will get many, many more peptides. If you are limited in material, though, what you want to do, you want to inject 100-time-fold less protein, and we get similar results as we were having in the microflow configuration. So practically, what does this mean? Because this is peptides, what does this mean for us, for our field? We do typically experiments in the microflow and asking people to bring us 50 to 100 micrograms of protein. Sometimes, if you're talking to a structural biology lab, they have no problem giving you that. But if you don't talk to a structural biology lab and you want to identify big complexes and investigate those, they are very limited in amounts. So we are super excited because now we can complete the full time course using 2 micrograms of protein per time course. So again, this is great. We felt really hopeful, and over the past 6 months, we operate the system now for 8 months. It's very robust and reproducible. And again, I would urge you to go to Shonak's talk tomorrow because he shows more technical stuff on this. And next slide, I am showing some of the applications that we have been up to in the past 6 months. So we have been using diverse ligands, and we have been analyzing all sorts of interactions, whether it's like small molecules, nanobodies, cyclic peptides, etc. So we have some systems for which an X-ray structure already exists, and these for us are serving as quality control measurements, as is the case at the top left, where you see the cyclin T, CDK9. We can identify, in blue I am showing regions that are impacted by ligand binding and show protection when you label the sample. So you can identify the binding site, and another nice thing that you can do with the technique is also see allosteric effects that you typically lose when you do static structures. So that's great. Also, we can do epitope mapping. That's the bread and butter in our field. And at the Broad, we have a very big generation platform for nanobodies. So this is something that we do almost on a daily basis. We can also do cyclic peptides. In the case where we don't have structure, as I'm showing to the right, we find a region which is a probable binding site, and then we give it to the structural biologist to take it from there, and we can do it with fragments and amino acids. So we can accommodate in this technique a wide range of affinities from nanomolar to micromolar, and we can even add millimolar amounts for low affinity interactions. And now we have tested it extensively, and all of our time courses have been completed using minimal amount of protein. Okay. So switching gears now, I hope all of you have seen the Orbit Trapezidion Pro in this conference that Thermo recently launched. We have a long time collaboration with Yuji, and we are really grateful because he gave us early access to this instrument and to evaluate its performance. So for this, we wanted to take a protein that has been characterized really well and it's of interest to the public. In this case, it's KRAS. And its ligand adagrasib. As you all know, it's FDA approved, it's a covalent inhibitor, and it targets the KRAS G12C. So this interaction has been really well characterized, and it has been shown that adagrasib binds in the binding pocket of switch 2 region. So the way we set up the experiment, we have the protein with DMSO, then we have the protein with a ligand, and we perform a time course experiment at 20 degrees Celsius. And we wanted to test 3 different levels. We wanted to test HTX at the intact level, then at the bottom-up level, peptide level, and then we also wanted to test peptide level with ETD, because if you don't have a high-resolution structure, the question that we keep getting from people is, can you find me binding residues? And you can do this if you use like a low-energy technique, in this case, ETD. So why would you want to do it at the intact level? It's really high throughput. It's a matter of throughput. So for us, especially when we have multiple ligands, this we can say immediately whether a drug binds or not without going to the peptide level information. And this saves us a lot of time. So when you do high throughput intact, you use 3 minutes gradient, so you can actually have 150 injections per day. And the answer here is like, does my drug bind? Yes, no, moving on. Also, the intact gives you an idea of your experimental conditions. So many times you don't see a change, but it doesn't necessarily mean that there is no change. It could mean that you're not sampling the correct time course window, or that your temperature is not correct, and you need to play around with these conditions. And this is totally dependent on the protein and the structure that you have. And this is, again, time-consuming if you do it at the peptide level, because you have thousands of peptides, whereas in the intact level, you just have to deconvolute the mass and get a result. So what I'm showing to the right is the difference in the deuterium uptake we observe between the DMSO state and the adagrasib state. What you can learn from this is how many solvent-accessible residues you have at these conditions, in this case 25 Dalton, in the case of DMSO, and you see a constant change of 7 Dalton for the first couple of time points, and the difference actually gets bigger as time passes. So that, for us, is indicative that adagrasib causes some stabilization of the protein. So it's a global view of how the protein moves. So we can learn from this, and we really love this. Then what you want to do is you want to ask the next question, where does my ligand bind? And this is when we go and do peptide level work, the bottom up. So what I'm showing here is the 4 time points again. This is a slightly different graph. On the y-axis now, we have a difference in the deuterium uptake of DMSO and adagrasib. So each line represents a peptide, and the x-axis is from the N-terminus of the protein to the C-terminus of the protein. This is what we call a Gooch plot. So what you see here is a clear protection in the P-loop switch 1 and switch 2 regions at 20 seconds, and nothing else. At 100 seconds, you start seeing this alpha-3 helix showing a difference, and by 1,000 seconds and to an hour, you're seeing this alpha helix picking up more deuterium and more. And at the bottom, I have this mapped on the crystal structure, and it makes total sense. And now you can see again how your protein moves and get this local information that is really crucial for downstream experiments. And this is also highlights why you need the time course experiment. If you had done only the 20-second time point, you wouldn't be able to probe what happens to the alpha-3 helix. So you need to select a few time points and do this because there is a lot to learn. So, okay, now we know and we are checking, we have a covalent ligand here and we have a structure, so that's great. What happens if you don't have a structure and you want to enhance the resolution even more from the peptide level? Because it's possible here with that many overlapping peptides to get down to 2-3 residue level resolution. And the field has come a long way. If you have seen peptide maps from the past, you had 100 peptides. Now it's typically we get like 1,000 to 2,000 peptides. So that's a great way of doing the experiment. However, what if you need to take it further? And this is where you want to employ ETD fragmentation. Now, why ETD and not HCD or CID? Work from the Jorgensen lab up in Thomas Jorgensen in Denmark, which was also the lab that I was trained. They showed that if you want to prevent what we call deuterium scrambling on the peptide backbone, you need to use a low fragmentation technique. And what do I mean by scrambling? They came up with this model peptide, P2, that you see it has a very basic N-terminus, and then the isoleucines and lysines. So under non-scrambling conditions, you should be able to measure loss of deuterium from the N-terminus. Histidines exchange deuterium really fast, back exchange deuterium really fast, and all the deuterium should be kept on the C-terminus of the peptide. So for people who are in the field, they know really well that this is the first experiment they need to do to optimize the optics in the system and the temperatures, etc. So what Yuji has done here, she has run the model peptide in the Excidion Pro, and we see with the black curve that there is no scrambling. So you see the green line is the theoretical curve. What happens if you have what happens if you don't have scrambling, and we see a very low degree of scrambling. So it works great in our hands. So how do we go about it? We select few peptides because analysis, I will say, it's very time consuming. So you cannot go and do ETD on all your peptides. What you want to do is you want to select peptides of interest. In this case, we have selected peptides from the 20-second time point and the 1,000-second time point, 76 in total. And we have generated an inclusion list, and we do targeted ETD. And I have to say that this data were acquired last week. The analysis is very tricky, it takes time. So please, I don't have results from this, but I want to show you what would be the perfect ending to the story. And we are wrapping up the paper and the analysis. Someone is working really hard on the background back in Boston to finish this, and we should have these results soon. So to summarize, at the Broad, we have a novel nano-HDX platform, and it really expands the possibilities in structural proteomics. The sensitivity that we have now, the throughput that we can do, and the resolution we have renders this an excellent tool in drug discovery for multiple applications, either screening, using minimum amounts of protein, even like for fragment libraries that you don't have enough compound. If you scale proportionally, then you need just a little bit of this. And again, proteins with limited sample availability is a very strong point of the nano-HDXMS. And the ATD capabilities on the Orbitrap Excidion are amazing. We are very much looking forward into it. Another thing that I didn't mention is that with the Vanquish system, you can actually, what I like about it, you can go from micro to capillary to nano with minimum changes in configuration. So what Thermo has tried to do is bring it all in one, and we are super excited for this also tool because it's working really great for us. Last but not least, I would like to thank, first of all, Shonak in our lab, who is giving the talk tomorrow. He's back there, so please go talk to him if you want to learn more about it. He has been working really hard over the past year to get this going. And of course, Yuji at Thermo. She has been a great collaborator and friend at this point, and she's presenting a poster around this today, so please go and talk to her as well. The CAR group, again, we are a very big proteomics group. We are very fortunate because we get inspired by things that you all guys do, and then we apply them to our field. So if you have interesting projects, we would love to hear from you. We have many different types of collaborations, so please come find us. Thank you. Okay, not bad, right?
Thanks, Malvina, for the great talk. Let's give another round of applause to her. And if you have any questions, we will have the group Q&A in the end. And just to remind, in case you have two purposes for this workshop, one is to hearing about science, another one is to collect the building blocks. Please wait till the end. There is a QR code. Scan the QR code. You will get what you want. Okay, our second speaker, Varun Gadkari. Professor Varun Gadkari earned his bachelor and PhD degree in biochemistry from the Ohio State University. He was a postdoc researcher in the lab of Professor Brandon Rotolo at the University of Michigan, where he advanced and applied native mass spec method and studies various interesting systems such as intrinsically disordered proteins, chaperones, biotherapeutics, and non-coding RNA. Varun joined the Department of Chemistry at the University of Minnesota in 2022 as an assistant professor. His research group develops and applies native mass spec method to investigate biomolecular structure and stability. Professor Gadkari is particularly interested in charge detection and ion mobility. His group is advancing this method to tackle long-standing analytical challenges in biomolecular characterization. And he won some awards. He was recognized in 2020 by ASMS as an emerging talent in academia and was awarded an ASMS Research Award yesterday. Congratulations, and let's welcome Varun.
Thanks, Weijing. And thank you all for being here this morning. I know it's early, it's the third day, so really grateful that you're all here. So I'm going to take this time this morning to tell you about some exciting work that we've been doing in my group. We were one of the first groups to really jump on the direct mass technology trend, but our application case right from day one was always going to be mRNA. So I'm excited to show what we've been working on for the last year. Before we get going, I'll add a lot of this work is made possible by tremendous help from Weijing Liu, who's been taking data with us, as well as Kyle Bowen, who's not here, but really grateful to you all for that support. So, sorry. We'll do a quick primer, which I think doesn't require a lot of explanation these days, but following the COVID pandemic, we've had a broad adoption of mRNA vaccine, and it's well understood that these medicines kind of brought us out of a global crisis. And where we stand today is that, you know, at least 82% of Americans have at least one vaccine, and 67% of the world has some series of a vaccine, and about a somewhat higher percent has at least one shot. So there's a lot of statistics on the World Health Organization dashboard about this stuff. Where this leads us, though, is that we've kind of emerged into this era of nucleic acid medicine being a lot more accessible and acceptable. And I think while many people in this room may have been researching this for decades, I think we're happy to see this kind of in the spotlight. And so there are many companies now developing many medications and vaccines and therapeutics in this space. I've put up two that most of us are familiar with. And just between the two of them, they have approximately 60 mRNA-based therapeutics in development right now in their pipelines. And one of the big things that comes to play now is that they want to look at really important quality control attributes of these molecules before they send them out to you and I to take in case we need them. And so mRNAs are the RNAs that we'll focus on today, but of course there are various nucleic acid medications out there, therapeutics out there in development, are just large and complex biomolecules. If you think about the fact that your proteome usually has an average monomer protein mass of about 40 kilodaltons, and that every one of the amino acids in that protein requires 3 bases, you're looking at some of the largest biopolymers in nature, okay? And so these are just inherently larger biomolecules, and they're pretty heterogeneous. As with proteins, mRNAs also have critical regions that are involved with very important, aspects of their function, whether that's as a therapeutic or just in your body in vivo. And so we're looking at some important aspects in terms of things like the 5' cap, which is involved in preventing degradation and some sorts of control of expression. You have the UTRs, which are often structural regions that are going to heavily impact things like expression of these mRNAs. We have the open reading frame, which is effectively the payload of these molecules, whether, again, in a therapeutic or an in vivo sense. And then we have the poly A tails, which are, again, critical for preventing degradation, but I think a lot of analytical groups will tell you that these are also the part of the molecule that give them a lot of trouble in terms of characterization. So in terms of heterogeneity, we can have inconsistent poly A tails, we can have inefficiencies in 5' capping, all which lead to downstream issues like truncation and so on. And so this heterogeneity, if we have to boil it down to 3 things, comes from things like that heterogeneity in those poly A tails, things like that. High levels of counter-ion induction. These are also the most heavily negatively charged molecules that you have. There's a negative charge between every single base, and so these are going to be heavily adducted with magnesium, sodium, or practically any cation that they can get a hold of. And so the need, to boil it down really, really simply, right, and this is not all-encompassing, is that we need rapid sensitive analytical methods, which will allow us to measure several attributes at once and learn something more than maybe what we can with our existing methods. And given that we're at ASMS, of course, we believe the solution here is mass spectrometry. And so we'll start with a quick primer, and I like this slide. I'll admit I stole it from Wei Jing, that mass spectrometry is a misnomer, and we only measure M over Z, right? We call it mass spectrometry, but with all of our spectrum, the x-axis usually says M over Z. And so we can take a spectrum like this, an electrospray ionization mass spectrum, and we have to deconvolve it to get an intact mass. This is an example of a protein, carbonic anhydrase, and so we have a charge state envelope. And the 2 ways we do this, right, we take either a close look at a single peak, and if we're lucky, because many of us use Orbitraps, we have isotopic resolution on these peaks, and then we see the spacing between these peaks, and we do a little simple math, and we figure out that the peak that's highlighted is a 10 plus, and we annotate the rest, and we get some confidence in our deconvolved mass. If we're using a TOF, like some of the other work we do in my group, we don't always get the isotopic resolution on a single peak, but we do have adjacent peaks, and we can use the adjacent peaks to, again, do some pretty simple arithmetic, figure out our charge state is, of course, 10, assign the rest, deconvolved. So we can measure mass pretty well from ESI, deconvoluted peaks, and so on. The problem starts when we start looking at things that are very big or very heterogeneous, all right? So we've all seen these peaks at the top, these denatured spectra of proteins. You get these low M over Z, high intensity, well-spaced charge states. Follow what I just said, figure out the deconvolved mass, easy. Native mass spectra, a lot more common these days. The structural biology folks in the room are not unfamiliar with this. We move up the M over Z scale, but we still have well-resolved ESI peaks. Again, we can deconvolve that mass. The conventional image here that I'm showing talks about a high mass resolution problem, right? You get really into the megadalton range, your ESI peaks run together, no deconvolution possible. But this problem also occurs when you're measuring things that are just only about 300 kilodaltons if they're really heterogeneous. We've seen some data actually probably at this meeting in this room maybe 3 years ago talking about Enbrel and DMT, right? Direct mass technology. And so I'll show you our interest, which is mRNA. And so what you're looking at the right are 4 mRNAs that I'll be talking to you about today. And we were looking for a set of mRNAs, and the first thing we did was we took native mass spectra. Now, my hypothesis was that all 4 of these mass spectra would look like the 2 on top, unresolvable humps, but Orbitraps are good analyzers, and so we ended up getting some resolution on the peaks on the bottom 2. And so what you're looking at here is the mRNA for encoding the proteins EGFP, F-luciferase, beta-galactosidase, and Cas9. You can see that the bottom 2 have some resolution on those charge states. We did perform a deconvolution. We can get a mass number. Somebody who's been doing native mass spectrometry for about 10, 12 years, I wouldn't show those as confident assignments for a native mass spectrum because there's a lot of overlap in those peaks, and while the EGFP number is actually very close to the expected mass, I can tell you from the data that you'll see in a moment that there's a lot going on under there. As a last comparison, As a last comparison, I've blown up the EGFP mass spectrum on the left here, and under it, I've plotted in magenta the actual mass spectra we can get for GROEL, which is the 800 kilodalton, so about 2.5 times the size of EGFP, pure protein standard that we use in native mass spectrometry all the time. So those peaks are real, those are not just drawn in. That is how clean that data can look. So mRNAs at about 1/3 of the size are a lot more complicated. So our approach and our solution to this, and something that I was really excited about when I saw this in Philadelphia in 2021, was the ability to do charge detection mass spectrometry on an Orbitrap. I went home from that conference and immediately rethought what we were going to do as a group. And so direct mass technology has allowed us to tackle this problem of heterogeneity at the single particle level. We do this by operating our Orbitraps, as many of you already do. Your ions are injected. You're taking a time domain signal. But what we do this time is we look at the ions individually. Our frequency data still gives us the M over Z, but as we track the intensity of these individual ions over the time that we measure them in a single particle manner, we find out what the charge state is. And this is a intensity-dependent calibration that we perform. This method you might have heard of referred to as selective temporal overview of resonant ions, or STORY. And so if you hear me say story plots or story data, that's what I'm talking about. What that results in is being able to take that kind of hump of noise I showed you earlier and turning it into what looks like the barcodes in the middle spectrum here. And these barcodes are a wealth of information. These are individual ions for which we are measuring M over Z and Z directly. And when you have that, you're once again doing simple math and going straight to the spectrum at the end, which is mass. So to jump into the data and what we've been working on here, I wanted to talk about how we're measuring mRNAs. The challenge we faced when we came here was that we didn't have a standard. I couldn't find the NIST mRNA, right? There wasn't just something we could order that was well quality controlled and well understood. So we said, all right, we're going to start the field and set some standards. And if you all want to use them, great. If you don't, that's fine too. I do hear that NIST is producing an mRNA standard soon, and we should be hearing about it in a year or so. To validate this process, though, we had to check with a bunch of vendors, and we landed on TriLink Biotechnologies. They have, and this is not an endorsement, I don't work with them or anything, but what they have is 4 mRNAs that we really liked across the mass range that are well quality controlled and always available in about 2 days. So we order 100 micrograms at a time because we don't need a lot of sample, and so I've provided product numbers if you'd like to try these out. I'll say at this point, throughout the presentation, I might say EGFP, Cas9, beta-galactosidase, F-luciferase. I'm not referring to the protein, I'm referring to the mRNA that encodes this protein, okay? So we ordered these. They come in about 1 mg per ml concentration in a solution of 1 millimolar sodium citrate, and we dilute them in a 200 millimolar ammonium acetate prepared in RNase-free water down to 20 nanomolar. That is our sweet spot for concentration for analysis, and we proceed to measurements. This one bullet point is woefully understated. We spent a few weeks figuring out what the right condition was to prep this. We were happy to find out that the best condition is in fact just diluting. We can do that because we're measuring single ions and we have to be at low concentrations. And a lot of folks internally and externally come out ran some stuff for us at Thermo. We all had to try different things to figure out what was working. And of course, all of our measurements were performed on a Q-exactive ultra-high mass range, hybrid quadrupole Orbitrap, and this instrument has been able to really just unlock the ability to measure these in high resolution. Data acquisition is being performed in Freestyle, and processing is being performed in Storyboard, which is software available from Proteinaceous, as well as UnitXCD, the program being written by Michael Marty and his group, and they maintain that software, and they're very responsive as well. We're using nano-electrospray ionization. I won't go too much into detail with that, but the key thing in our measurements is that we're ionizing from ammonium acetate when we're doing native measurements, and we're ionizing from 50% methanol when we're doing denatured measurements. So let's look at some data. We started looking at mRNAs first in positive mode, if no other reason than that positive mode is easier. And while everybody thinks you have to look at them in negative mode, you in fact can look at them in positive mode, and I'll show you some good reasons to why we should keep doing that. So looking at that EGFP mRNA first, you all should remember, a few minutes ago I showed you how broad that distribution was and how, the values between those peaks were not that low. And that's because there's a lot going on under this mRNA's M over Z distribution. What we see in this initial measurement is that we have a main peak that's highlighted by that circle. That is the monomer mRNA. We pretty readily see a dimer, which is an impurity I think that most pharma customers would be interested in looking at and understanding if that's present. We also see a truncation that's spaced by about 50 kilodaltons, and there's actually a shoulder on the left side of that monomer peak as well. And so we see that there's a lot of character under that distribution that we can now see in a direct mass measurement. This also below it, I'm showing the M over Z versus charge distribution, and I've kind of changed the contrast so you can see some of the noise or the truncation signals, and you can really get a nice distribution, which I have a background in ion mobility, so I really like this two-dimensional representation of the data as well. Moving up the mass range to F luciferase, we see a pretty nice monomer peak with a notable truncation at -86. Interestingly, we never saw dimers with F luciferase, so we're starting to see that there are some things that are just properties of certain RNAs and not present in others. We then saw a monomer and a couple of different truncations for beta-galactosidase. We were starting to get worried at this point that we were producing these truncations, but we did some digging, and actually beta-galactosidase mRNA seems to have these regardless of where you get this RNA, and we started seeing some of this sort of data in people's data from CGE and things like that. So beta-galactosidase, the mRNA you get, will always have some truncations in it. And the Cas9, to put our mind at ease, it wasn't just a large mRNA problem. We get a pretty nice clean distribution of the Cas9 mRNA. And so all of these across the bottom show you the sequence mass and the measured mass, and you can see that we were hovering around 4% to 6% error on the smaller mRNAs and about 10.6 on the Cas9. And so this mass error is a little high for my comfort level, and so we were going to look into how we could do better here. Now, one thing to talk about here before we move into the discussion of being close to sequence mass is that I told you at the beginning that these are heterogeneous molecules and they're heavily adducted. We can do a lot of things to remove adducts, but we can't change the fact that despite the fact that there's a 120 poly A tail, as told to us by the manufacturer, that it actually is probably a 100 to 180 base poly A tail. There's always going to be that heterogeneity built in, so we're never going to hit an exact sequence mass, but we're going to get pretty close, and I'll show you how we do that. For sanity check, we did run the native mode data, so I'm showing 4 independent measurements plotted together, just for simplicity. And so in negative polarity, we see similar spectra. We see EGFP with a dimer present and some truncations. We see F luciferase. We see beta-galactosidase. Interestingly, the low mass truncations are less visible in negative mode, and these are some things that we have to start thinking about now when we analyze these things, that maybe, you have to look at things in both polarity. And then we see the Cas9, and the Cas9, we did see some dimer here as well. And so to bring up both sets of data side by side, you can see that they're actually pretty comparable in terms of accurate mass, and the errors are fairly comparable across polarities too. So if your target is accurate mass, you could probably get away with doing it in positive mode. So let's improve mass accuracy, and of course the benefit of this advantage is that we can do charge detection mass spectrometry, but we also have a collision cell on the end, and so that opens the doors to a lot of possibilities. And so we did the same measurements with our HDD voltage turned up. Now, in our native measurements, we have the HDD voltage on and we're using it, but we're using it to a point of optimizing transmission, getting a clean signal, but we're trying not to do any kind of inadvertent activation. But going for accurate mass, we turn up our HCD and we start looking at measurements that are now under 2% accuracy to our expected sequence mass of mRNA. We see a nice improvement in resolution. Actually, some of these have a mass resolution of about 35 or so. And so that gives us a pretty good sense that CDMS and collisional activation do in fact go hand in hand, and they're important to have. Cas9, the improvement comes in a couple percent. We're still struggling to get a nice accurate measurement on Cas9, but I'll be honest, there's a lot of things going on with these larger RNAs, and I don't know as much about the Cas9 mRNA as I do about the other 3. So we're still figuring out what we can do to improve our accuracy on this. We go ahead and tried a denaturing mode as well. Pretty common approach for doing accurate mass of biomolecules. So 50% methanol will clean up your mRNA pretty well. It'll desalt it, it'll unfold it. And we'll see charge state ranges from about 100 to 600. So we can definitely look at charge states in the 600, 700 range on this instrument. And I'll show you that at the end. We see comparable accuracy actually to the activated mode. About maybe, not as good, in fact. I would say the activated mode's better, but I can understand the simplicity of just preparing something in 50% methanol and spraying it. So it's still better than the native measurement by about 2-fold. And interestingly, the native mode measurement of Cas9 does actually come in close to about 5% accuracy. So we're now going to explore, maybe for the larger mRNAs, we have to transition to denaturing measurements if accurate mass is the key factor. I'm showing some bar charts here that kind of compare on top the native mode deviation from sequence mass and below the activated measurement deviation from sequence mass. Solid bars are positive mode, hash bars are negative mode. And you can see that you see about a twofold, threefold improvement in accuracy with activated measurements, and plotting them together, that becomes apparent. And so we're pretty confident that if you're going to do accurate mass measurements of mRNA, you should be using an activated mode in native or a denatured measurement. To give you the denatured measurement comparison, again, we do see an improvement in the deviation from expected sequence mass, and plotting them together with the HCD activated data, you see that they're comparable. And we're still making heads and tails of some of these things. We tend to see that the negative ions do better in denatured mode, the positive ions do better with an HCD activated approach. I'll be honest, this is a new field for us and for, I think, most of us. And so we're trying to understand really what drives this. To show you then my final case of why you should consider doing native. If you want to learn more than accurate mass, there is some benefit in taking a native approach. And so I'm going back to these M over Z versus charge plots. As I said, I'm from the mobility space, and so we love looking at shifts in these two-dimensional plots. And so if you look at the trend lines that can appear between these mRNAs, we start to define what we would call the Rayleigh line for RNAs. So basically, the charging of natively structured biomolecules as a function of their surface area as they get larger and larger. And I will be the first to admit a Rayleigh line based on 3 points is probably not like a truly a line. And we're trying to build this out. We have a lot of mRNAs at our disposal to do this. But what we started seeing right from our initial panel is that 3 of these fall pretty nicely on a line, but beta-galactosidase is kind of doing its own thing at all times. It's always hanging out with enough charges to support it being as big as Cas9, but actually it's about 500 kilodaltons smaller. So that tells us that the mRNA that encodes for beta-galactosidase is expanded in some manner or structurally more flexible. Now, you could imagine setting up a quality control assay around this, where if you have a product that you're working on and you know the charge distribution that it should land in, this is a good first step to look at it at the intact level. Does it fall in the charge state range I'd like, or does it not? If it does not, I will pass it on to further analysis. We see this happening with the activated sample also, so you can do the activated native measurement, get accurate mass and accurate kind of structural measurement to some degree. Here's the denaturing data. Note the change in axis on the charge states. We don't see that in the denaturing data. So kind of a final point here, right? I think the takeaway is native activated, regardless of polarity, is kind of the way to go here. So it really helps to have the ability to activate these ions, clean them up, get a better mass measurement, and understand what we're looking at. So kind of towards enabling these simple things, I talked to you a little bit about this dilute and shoot sample prep. We just dilute to 20 nanomolar, we head to the instrument, works out pretty well. The important thing with our method other than that, and this is something I'm really proud of, is that we were able to do this actually with very small amounts of changes. And I worked closely with Kyle Bone at Thermo on this and Wei Jing, that we said we should be able to translate this quickly. So most of the settings in this instrument are operated at default values. 3 regions are modified. The S-lens runs at 0. You can talk more about this with me or Wei Jing or other folks later, but we have a feeling that at higher settings, which are the default, we actually bias against higher mass analytes. The in-source trapping, which is a popular setting on this instrument, we actually find that we don't like to use it for mRNAs. We have it off for the low mass mRNAs, and we set it at 1 to 10 for the larger mRNAs. There's a hindrance of seeing these ions with that on, and it's a RNA, DNA specific thing that we're observing. And of course, the HCD cell, I mean, this is the powerhouse of this measurement. We can collisionally clean things. We can use different gases. There's no reason to say that we shouldn't have a collision cell in a CDMS instrument. So we're really excited about this, and this allows us to do so much more. We're going to do a lot more with this and understand how we can fragment these RNAs and learn more about them. And so in conclusion, we have established a set of readily available mRNA standards that I hope you'll all adopt if you want to make these measurements. The NIST standard that's coming is actually an F-luciferase standard, so we were on the right track with that. And so we should be able to do some interesting measurements soon. We're within 2% for small mRNAs, small in quotes, and within 5% for big ones. And we can see things like dimer populations and global structure. And so this is really valuable as a technique for looking at more than just accurate mass. And we've designed some optimized translatable tuning methods that are ready for broader adoption. So I hope that you'll look for this paper coming out later this summer and be able to take this to your own labs and be looking at some mRNAs or large nucleic acid molecules. With that, I'd like to thank my team. Rahana Afroz, who's highlighted in red, is the grad student who's been cranking out this data over the course of the last year. Every week there's something new. And the collaborators at Thermo Fisher Scientific, Kyle Bowen, who's making trips out to Minnesota just to run these samples with us, and Wei Jing, who's meeting with us to break down what the data looks like all the time, and Rosa, of course, for connecting us and getting us started on this pathway, have been instrumental in our success and our ability to do this in 1 year as opposed to what would have probably taken like 3 or 4 years for just us. So with that, thank you, and I'll pass it on to my next speaker.
Thanks, Varun. Very exciting results. Let's thank Varun again for the great talk. Our last speaker, Professor Fan Liu, cannot be here today for some very innovative reason, but we have another wonderful person, wonderful speaker, Philip Loso, who will deliver the talk about cross-linking. Philip is a senior VP of FC Biotechnology and leads FC's headquarters and Proteomics Innovation Center in Berlin. Philip graduated from Utrecht University, where he developed an integrative mass spectrometry approach to study protein structures, modifications, and interactions. Before moving to FC, Philip was an editor and later team manager at Nature Communications, overseeing manuscripts in all areas of mass spec, biochemistry, biophysics, and structural biology. Let's welcome Philip to the stage.
Thank you, Wei Jing, for the introduction. And I know it's hard to take surprises, especially at the early morning, so I hope you guys are all coping with seeing me here instead of Fan. She's certainly very sad not to be here. She would have loved to present some data to you guys, but yeah, I'm also excited to show you some of the recent work from the Leo Lab, and yeah, thanks to Wei Jing and Rosa and Thermo for being so flexible and accommodating that. So I guess this crowd doesn't really need an introduction to cross-linking, but just very briefly, cross-linking relies on using these small chemical molecules to that have two reactive groups that can, covalently connect to amino acid side chains. And these small cross-linkers, they have a fixed spacer arm, which means that only amino acid side chains at a certain distance will connected to each other. And in this way, you can freeze protein complexes in whichever biological system, be it in vitro, be it even intact cells, in situ, in space, and then you do a mass spec sample prep, you get cross-linked peptides, and you can identify these cross-linked peptides, and then in this way know where proteins are connected. So essentially you get distance constraints out of your sample, so you know which parts of your protein are close together, and this can tell you something about protein structures, about protein interactions, and actually in intact cells or intact organelles also something about protein localizations. And to get this out, you can use different softwares. X-Links is the one that Thermo offers, and it works very well. Of course, there are many more out there, and some of them I'm also going to mention to this talk. So, as said to you, you can get protein interactions out of there, and this is an example of an interaction network that you can get out of a cross-linking experiment. And you see, a very, very widespread network, and you see beautiful these protein neighborhoods, like different complexes, different compartments, different functional groups. And you also can appreciate that because you see direct connections between proteins, it becomes possible to distinguish between which proteins are actually interacting directly versus which are interacting indirectly. And more than that, you can also zoom into this network, and then you can see where your crosslinks are exactly located, and that gives you some information not only which proteins interact, but where these proteins interact. And now the cartoon here just shows a domain-level overview of where proteins interact. But of course, you can also imagine mapping these crosslinks onto crystal structures, cryo-EM structures, AlphaFold models, or you can also use them to guide your computational or experimental structural modeling. Now, Fan's lab has been invested in cross-linking method developments for many years, and I'm just going to show you three recent unpublished examples of what they've been doing to get this cross-linking workflow being more efficient. Now, the first example is about the sample preparation workflow, And the mainstay in Fan's lab is really this crosslinker, DSBSO, which has been developed over 10 years ago by Lan Huang's lab. And it's really a beautiful reagent, a very powerful molecule for several reasons. So on the one hand, you see it has two NHS ester groups, and those react specifically with lysines or protein N-termini. So we will connect these groups if they're close enough together. It has a spacer arm of 14 angstrom, which means, amino acid side chains in this distance can be connected. But most importantly, it has this azide residue that allows, this azide functionality that allows click chemistry enrichment of your crosslinks, and it has these two sulfoxide groups that allow cleavage in the gas phase, so by CID or HCD, and that will give you signature ions that simplify the identification of your crosslinks, but also make it possible to actually identify your crosslinks by searching for linear peptides, which makes it easier to look at proteome-wide databases. Now, this DSP is also a great reagent, but sample prep can take a minute. And now there's Pin Lien, who's actually just standing over there and who's going to give a talk in the next session. And he, although I'm not in Fan's lab directly, I know Pin Lien to be a very hardworking guy. But I guess at some point he was also tired of having like these multi-day sample preparation processes. So he tried to figure out a way, how can we actually do this in one day and then, call it a day fairly early and have a nice dinner and also maybe be able to watch a movie still. And so he really looked into every step of the DSBSO sample preparation process, and he improved the digestion by actually merging it with the DBCO enrichment step. He found several ways. To clean up the sample, so clean up unbound peptides, but also clean up some contaminants. He found new ways of fractionating the sample, and he also optimized LC gradient and HCD and the HCD methods to actually get these get better mass spectrometry data out of these DSBSO cross-linked samples. And unfortunately, I do not have enough time to talk to you about all of these steps. That's why I really invite you to go to the cross-linking session after this one and listen to Pin Lien's talk. Of course, he will be also much better able than me to tell you all the technical details about what he has been doing exactly. But just to give you an idea, I'm going to talk about the contaminants cleaning step. And so what Pin Lien noticed is, maybe I should go one step back. So what you do with DSBSO is, in order to enrich the crosslinks, you mobilize it on DBCO beads with click chemistry, and then you elute the cross-link peptides later by adding TFA. And so now what Pin Lien noticed is, if you increase the TFA concentration, you start to get like this gel-like residue at the bottom of your tubes. And we were wondering, well, he was wondering what that is, and he looked a bit into the chemistry, how this elution actually works. And so DBCO forms an acetal structure with the cross-link peptide, and that is then hydrolyzed with TFA. And interestingly, DBCO is immobilized on agarose beads, and so these agarose beads also form an acetal structure together with DBCO. So what happens if you actually elute with TFA is that you also cleave these DBCO agarose conjugates, and as a result, you will get some of this dissolved agarose forming this gel-like thing, and this is actually a non-peptide contaminant, which has been shown before that there are these non-peptide contaminants that can compromise your cross-link identification. And so Pin Lien was thinking, maybe this is probably the main non-peptide contaminant we're looking at, so how can we get rid of it? And so he has been looking into offline SEX-based fractionation or cleanup to get rid of this sample, to get rid of this contaminant. And the first thing he did, he just did this single-step elution with 50 and then 500 millimolar ammonium acetate, and that actually already worked like a charm to get rid of this non-peptide contaminants. However, then we thought, why can't we take this maybe one step further? Why only cleanup? Why not also try to do this use this as a fractionation step, since we are doing this SCS-based stage tip anyways? And of course, SEX fractionation for crosslinking is not unheard of. Often you do this online and you use sodium chloride. It's not volatile. So we thought, why not try to do this offline with ammonium acetate? because that's, of course, volatile and so much more compatible with mass spec. And so Pinion did this with a hex cell sample, and the results were really quite impressive because he got amazing identification numbers already in every individual elution fraction. And then if you look at the fractions cumulatively, you really have a massive increase in identifications on all levels, like CSMs, residue pairs, and also protein-protein interactions compared to the single-step elution. And looking a bit more into the individual fractions, so this plot shows differently charged cross-linked peptides, +4, +5, +6, and how they elute across these 7 fractions. And so what we see is basically what we were hoping to see, that higher charged peptides would elute later at later fractions, but also that the majority of peptides actually only elutes over 1 or 2 fractions. So which means that this offline fractionation, and even though we use ammonium acetate rather than sodium chloride, has a quite decent separation. So again, this is one step. In the end, did a lot more. But after he had done all of this, he just looked at the interaction networks he gets. And so he worked with hex cells, but also with human mitochondria and bacillus samples. And I will say, for hex cells, 1100 ppis, I would say you can still get a lot deeper if you do deeper fractionation, if you if you use more input amount of sample. But with human mitochondria and also with bacillus, these are really decent interaction networks. We published a paper last year in Nature Communication with DSSO cross-linking and really extensive fractionation, where we essentially got the same, actually even a bit lower PPI coverage compared to this one. And this is really extremely fast. So I showed you how long the sample prep takes. It's really just a working day. And then also considering LCMS and cross-link search, this whole thing takes 2 days. We also don't need a whole lot of sample. This is 8 microgram input for the SCX stage chip. And these data were acquired on the Explorers and with 3-hour gradients. So the mass spec time within these 2 days was 21 hours. So we think that's really an efficient approach to move cross-linking to a higher throughput, more screening-like applications. Now, of course, this was still with the Explorers, but then last year, the first Astral mass spectrometer moved into the LEO lab, and they were very excited to test it and see how much this can add. And this is work done by Puka, another PhD student in Fan's lab. And the first thing he did is he looked how different gradient lengths and different loading amounts actually affect the performance or identification performance here. And the first thing I want to point to is if you look at the 120 and 180 minute gradient, you actually don't see a whole lot of difference in identification numbers anymore. So if you just look back at Pinlian's stuff on the Explorers, he needed to use 180 minutes. Here, we can easily get away with 120 just because of the higher sensitivity and the better performance of the Astral. On the other hand, we think even the performance at 60 minutes is very decent. And the good thing of 60 minutes is it's more compatible to lower sample amounts. So we actually start using these 60 minutes methods much more often now. And you can see this also here. This is an independent experiment. And so this is different gradients, and then also for the different gradient lengths, different sample amounts loaded. And of course, there's a trade-off. So if you have 60 minutes, if you only have a 60-minute gradient, you will identify less. But if you are mainly interested in getting an interaction network, knowing which proteins interact, and if you want to go for high throughput, then 60 minutes is definitely an option. However, if you want to get deeper coverage, for instance, for structural application, and you really need high-density cross-link information for the individual proteins, then maybe it's worth to go to longer gradients, like 120 minutes. The other thing that Puka found is, so what's not shown on this slide, maybe the most important message not shown on this slide, FAMES is always good to use. So we always see that FAMES helps a lot in terms of identification compared to no FAMES. However, Puka also looked at 3 CV FAMES versus 2 CV FAMES, and he found that 3 CV, so with 3 compensation voltages, kept outperforming 2 CV FAMES. So that's also on a human, on a hex cell sample. And the interesting thing here is, so 3 CV FAMES occasionally also performed better for us on the Explorers, but it certainly did not perform better on a 60-minute gradient, because then, it was just too slow, and the triggering of MS2 events would be compromised. However, because the Astral is generally faster, we see that even at these short gradients, speed is not really an issue anymore, and we still benefit from having these 3 CV FAMES and being able to access more features to be triggered. On the other hand, so I said before, FAMES is always better than no FAMES. So we were also curious to compare FAMES versus no FAMES on the MS2 level. And one thing that we found is, so 3CV FAMES and 2CV FAMES doesn't make a big difference here, but FAMES versus no FAMES makes a big difference in terms of the number of peaks in each MS2. And I mean, normally having a high number of peaks in MS2 is not a bad thing, but so our interpretation is, if you look at the reduced number of peaks when you use FAMES, together with the fact that we can actually identify more CSMs when we use FAMES, is that FAMES probably helps us to get rid of some precursor co-isolation. So some contaminant signals that actually come from co-isolated precursors that will then make it more difficult for the software to make a proper identification. Okay, so and then just very briefly, the last bit of our story. Very recently, we were able to get some data on the Astral Zoom, and that's also thanks to Rosa, who helped us acquire them. And this is really, very much work in progress, and even the data analysis is still ongoing, so I can just show you some vignettes here. And the first thing we were really excited about is just how rich these MS2 spectra look like. So this is also with FAMES, done with FAMES. And then if we, and you see, you can appreciate what a great sequence coverage we reach for cross-linked peptides and just how many signals we see in the MS2. And in fact, we get 3 times more ions in the MS2 spectra, even with FAMES enabled, compared to in the Astral Zoom compared to the Astral. And so this also is a challenge for software in the end. Like, we need to tweak our cross-link search engine, our cross-link identification softwares to be able to deal with these very rich data. And so FAN has been working a lot with Simin He's group and Peng Mao at the Chinese Academy of Sciences to update P-Link 3 to actually get it to work on Astral and Astral Zoom data. And so one first experiment we did in this space is, again, we used actually something that FAN's lab was developing together with APSI last year. What we did is we made a ground truth for cross-linking mass spectrometry. So we used recombinant proteins from APSI, we mixed them in a very controlled fashion and cross-linked them so that we would know exactly which proteins can and cannot interact. And this was published in Nature Methods last year. And now what FAN did is she just used part of this ground truth standard and spiked it into an E. coli, into a cross-linked E. coli lysate and searched the whole thing with P-Link 3 and used different ratios of this human standard compared to E. coli. And so what we see, of course, as expected, is if you spike in more human sample, you will identify more human sample and you will have less E. coli, and the other way around. At the same time, what we see is we have a very low empirical false discovery rate across the board. So we also think the software is sort of getting there to at least to be reliable in their identification. And I should also say here, this is a single shot experiment, this is a 60-minute gradient, this is a low sample amount, we still get very decent identification numbers. So we are very excited about the potential that the Astral Zoom has in terms of sensitivity, in terms of information richness, in terms of speed. And we think then the next step is really, get Pin-Lien's workflow combined with Astral Zoom so that we can also even make this even faster, take advantage of the instrument developments, and also keep pushing the software further so it can handle Astral Zoom data even better, and we can really leverage the full power that this instrument gives and also identify then more crosslinks from less samples and in shorter times. And with that, I'm at the end of my talk. I would like to thank Thermo, of course, especially Wei Jing and Rosa for helping us to make this quick switch possible. I want to thank FAN. I guess FAN wouldn't be thanking herself if she was here, but let's thank her since she's absent. And of course, her whole lab, especially Pin-Lien, Puka, and then the collaborators at CAS, especially Peng Zhi, who has been helping us to analyze this Astral Zoom data. And thanks all of you for your time.
Thank you, Philip. Another round of applause for him. And we have a few minutes for the Q&A session. Let's welcome all the speakers onto the stage.